Hey everyone, welcome back for the third of our wonderful sessions on seed ecology and evolutionary biology. Just another reminder that you should feel free to be putting all sorts of comments and questions in our Q&A as well as in the discussion forum, and that if you don't get a chance to get your question answered during the session, feel free to contact the presenters in the hub through the portal at any time. We're going to kick off now with a talk from Jose Barrerro on cowpeas.
Thank you very much, Adrienne, and good afternoon, everybody from Australia. It's a great pleasure to be presenting my work in this forum a year later. So I'd like to talk today about a biotechnology approach we are using to fix one big problem caused by a storage pest, bruchids, in a major crop in Africa, that is cowpea. And you will see how my talk, I think, it fits very well with this year's conference motto, this linking seed with needs. So why we work with cowpea? Because it's a key legume in West Africa. It was domesticated in this part of the world where there is a huge diversity, as you can see from this photography. Then it's a major source of protein for many millions of people every day. It is a crop that is tolerant to sandy soils and drought. It's a multi-purpose crop. It can be used to feed animals. It's consumed by humans. It's also a cash crop. But there's a problem, and is that the demand for these crops really exceeds the supply. And one of the main reasons for that is that there are some major insects that constrain the production and yields in this species. So there are two big major problems, two pests that affect the crop at different stage. One is the pod borer. This is a lepidopteran that burrows into the flowers and eats the fruits when still green. The other problem is caused during storage, and is caused by this Callosobruchus weevil, a coleopteran, that lay the eggs on the seeds. During storage, the larvae burrows into the seed and makes this big hole when it comes out as an adult. For the first problem, the pod borer, we have already developed a solution. We have developed a variety that is resistant to this pest. This is what the caterpillar of this Maruca species, the damage that makes on the leaves of the plants. And you can see this is the yield produced by a plant that is heavily infected by this caterpillar. However, we have created a transgenic Bt variety that carries this one cry1AB gene. And you can see here, it's totally immune to predation by this insect. The leaves are totally intact, and you can see the difference in yield between the plant that is protected in comparison with the plant that is not protected. We are proud to say that we have developed this product in the lab and taken all the way to the farms in Africa. And I show you here the certificate of permit to use this transgenic variety in Nigeria. And the project is going very well, where this year there is some bulking happening there in the farms in Nigeria. In this photo, I'm showing you a bulking farm in this place called Adamawa in Nigeria. The photo was taken in August 2020. And it's just an exercise to bulk up this new variety Sampe 20T that carries our gene. And it will be available for farmers from this year. For the second project, the second problem is these bruchids, as I say, cause major damage and economical losses. And it has it produces many problems. The first thing is that the grain cannot be sold or stored for very long. The seed viability is reduced. And also, it's the reason why we find in Africa high levels of pesticides in dry grains in the market. Can we solve the problem? Well, the thing is that there are very low levels of natural resistance to this pest, and traditional methods like acids or oils are really inefficient. And also, when people try to use chemicals against this insect, it's a very, very dangerous practice because the people are not very well trained to use these chemicals and don't follow instructions. So it's a very high risk to health. There are some solutions that have been implemented, like these big bags. It's hermetic bags that suffocate the insect, but it's not that useful for large amounts of seeds, for small amounts of seeds. So we want to use biotechnology. And there is already a tool that nature has given us, that is the alpha amylase inhibitor gene from common beans, from Phaseolus vulgaris. So this species in America, they produce inside the seeds naturally an alpha amylase inhibitor, a protein that stops the alpha amylase enzymes, and actually is even used in some formulations for some products against diabetes or for losing weight in humans. So this enzyme alpha amylase inhibitor is very useful in some part of the world. In America, some of the species of coleopterans there have become insensitive, resistant to these alpha amylase inhibitors and protein. However, in the old world, species like the one we're worrying about, Calosobruchus maculatus, still are sensitive to this thing. So we want to use biotechnology to move this naturally occurring protein from America to Asia or to Africa, where then it will be a useful tool to fight against these pests. We use this with biotechnology. We have cloned the alpha amylase inhibitor gene controlled by the own promoter in beans in a binary vector for transformation, and we have tested in a number of species. We have used this in field peas, and you can see the non-transgenic control, how has been damaged by these bruchids. And in the other side of the slide, you can see a resistant plant, a resistant transgenic plant. Similarly, with chickpeas, we have had the same results, where transgenic varieties are fully protected from the damage of these insects. You still can see the eggs on the surface of the seeds, but the larvae cannot emerge from them and doesn't cause any more problems. In cowpea, we have also developed a number of transgenic lines that are overexpressing this protein at a range of concentrations. So in this Western, you see here, these bands indicate the amount of protein present in the normal beans. Then you got a line of non-transgenic cowpea that doesn't show any of this protein. And then a number 6 of cowpea transgenic lines that show a range of expression of the protein. We test them in the lab, and we infested the seed from these transgenic lines and the control line, and then we measure the number of days that the adults need to emerge from the seeds. In the non-transgenic control, this is 26 days. In the transgenic lines, it's more than 120 when we stop the experiment. So we also develop a system of artificial seeds in which we can test the right concentration of the alpha amylase inhibitor that we need, and then we can choose the right transgenic line that has just enough. So the conclusions are that we have proven that this alpha amylase gene is very useful against bruchids in a number of species. We have proven that in the lab, and we think this alpha amylase inhibitor cowpea will generate a significant economic and health impacts in Africa. There are a few challenges ahead. We need to go through of course, biosafety and regulatory pathways with the proper allergenicity tests. And the final idea will be combine this alpha amylase inhibitor cowpea with our poor border resistant lines. And this photo is from an open field in an area where the transgenic lines have been presented to farmers. Thank you to all of our collaborations in Africa and in US, and my team at CSIRO, of course. And I like just to mention that this is this project that we are doing at CSIRO is one of the major contributions from Australian science towards achieving this sustainable developmental goal from the United Nations that Australia and other countries need to address by 2030. And that's all. Thank you very much for listening. I'll leave you here a couple of links in case you are more interested in learning more, and my contact details, you got any further question. Thanks for listening.
Good afternoon. My name's Sean Walsh from the University of Melbourne. I'm going to be talking today about variation in germination behaviour of physically dormant seeds extracted from forest soil samples. This is part of a PhD project on fire and seed bank dynamics. So, it's easy to study the behaviour. In this project we're interested in seeds that have been in the soil for some time. And the aims are to build a model of the effect of heat treatment on germination all the way up to lethal levels, to quantify the time to 50% germination, and to examine site-to-site variation in germination behaviour. My target species is Acacia dillbata, silver wattle, which is very common in southeast Australia. And my study area is the Western Central Highlands of Victoria, and I took soil samples from underneath pure stands of Acacia dillbata trees. So why study site-to-site variation in germination behaviour? Well, this can provide really useful insights into seed ecology, although many mechanisms could be involved, and some of those are listed here. And why extract seeds from soil? Well, we can study the germination behaviour of soil-stored seeds by applying heat treatment directly to soil samples, then laying out the treated soil in trays in a glasshouse. But using this method to study the full thermal response of seeds from many sites would require a huge amount of soil and glasshouse space. On the other hand, if you take the seeds out of the soil, you can design a more precise experiment that takes advantage of the number of seeds that you find at each field site. And it also allows observations of extra things like seed mass and seed morphology. Looking now at a bit of theory, I needed a function to represent both the dormancy release phase and the seed mortality phase of a germination response. And I didn't find any such function in the literature. So I combined two logistic curves to create the function you see here. And each parameter of this model has a physical meaning, and we'll be looking at some of those parameters shortly. Quick overview of methods. Acacia seeds were taken out of soil samples. And based on the number of seeds that were found at each site, a custom experiment was designed. Heat treatments were applied, then seeds were germinated in a standard growth chamber. And germinates were removed one by one as they emerged and grown on to confirm species ID. Starting with 160 kilos of soil, I took out about 3,000 acacia seeds. Then an aspirator was used to discard any low-density seeds, and batches were inspected under a microscope to check for any non-target acacia species. Then a germination experiment was designed with a nonlinear Bayesian power analysis to reduce bias and uncertainty in fitted model parameters. And the final design involved a subset of my field sites, treatment temperatures from 40 to 140 degrees Celsius, with unheated controls, and about 1,400 seeds were used in the experiment. Some of this work had to be done from home during a coronavirus lockdown, but that actually worked out really well because it meant that I could keep a close eye on the experiment every day. Now looking at some results. Here I plotted the final germination fraction as a function of treatment temperature, with the different colours representing seeds from different sites. And this blue curve here represents the fitted model. Now zooming in on some of the model parameters. This parameter Gmin is the non-dormant fraction. So these are seeds that don't need any heat treatment in order to germinate. And it was only 4% in this experiment, clearly indicating that the vast majority of the seeds from my samples were physically dormant. The next parameter is Gmax, representing the maximum possible germination under the constraints of the current experiment. And this was a little over 98%, again, indicating in this case that the seeds that I obtained from soil had very high viability. Looking at two more parameters now. On the left we have the dormancy release threshold temperature, which came in at 63 degrees Celsius. And on the right we have the mortality transition temperature, which came in at 115 degrees Celsius. As you can see from these error bars here, the parameter uncertainty in the mortality threshold temperature is quite a lot less than the parameter uncertainty in the dormancy release threshold temperature. And I thought this was very interesting. So I looked at this from another perspective, and I refitted the model on individual sites using a subset of sites where I had enough temperature treatments to fit the entire curve. And as you can see, the site-to-site variability in the mortality threshold is again quite a bit lower than the site-to-site variability in the dormancy release threshold. So why might this be the case? Well, the dormancy release process involves opening a tiny water gap in response to heat. So small variations in that gap process could easily allow significant variation in the dormancy release threshold temperature. On the other hand, in terms of the mortality process, acacia seeds do have some defences against extreme heat, but these are quite limited. So small variations in seed morphology or even the physiology of the seed, and some of that's things like heat shock proteins, probably have very little effect on the mortality threshold temperature. Looking now at some time to 50% germination results. Here I've plotted the germination fraction as a time series as the experiment progressed, with the different colours representing seeds from different sites. As you can see, there's a fair bit of variation between sites in terms of the speed of germination. Now on this graph I've plotted the time to 50% germination as a function of the forest stand age. And although there's a fair bit of scatter here, there is an indication that seeds from older sites germinate faster. And fitting a regression model, we ended up with a p-value of 0.025. So why might this be the case? Well, the faster germination of seeds from older stands could be a result of the age of the seeds themselves, as we know that there are changes in the seed coat when you bury seeds in soil and watch how that changes over time. But it could also be to do with the age of the trees themselves. So we know that very old trees are successful survivors. So there could be a population fitness advantage if seeds from older trees germinate earlier than other seeds in the soil. And this has been demonstrated in another acacia species, Acacia melanoxylon, by Cruz et al. 2017. Summing up these results, Acacia dilbata seeds found in forest soil had very high viability and low non-dormant fraction. The seed mortality transition temperature was 115 degrees Celsius with not much variation among sites, suggesting limited plasticity and scope for adaptation in response to extreme fire. And seeds from older stands germinated faster, which could be related to seed age or maternal tree age, or perhaps both. More generally, a new function with meaningful parameters captured the full thermal response of physically dormant seeds. And this function is well suited for use in a mechanistic model. And manual extraction of seeds from soil allowed a detailed study of site-to-site variation in germination behaviour of soil-stored seeds with a minimum of field and lab costs. I'd like to thank all the people who assisted with this work and my funding sources. And thanks very much for your attention.
Hello. Today I'll be talking about seed microbiomes of native Australian plants and how this knowledge can be used to inform restoration practices. I'll present a brief overview of the field and discuss the work that I have done through the first year of my PhD. We all know of the human microbiome, but we may not know about a lesser known microbiome, the plant microbiome. This microbiome is comprised of bacteria, fungi, and archaea that exist both epiphytically, that is on the outside of the plant, and endophytically, that is within the living tissues of a host plant. And endophytes have been shown to exist in all the different organs of a plant, but we know the least about those within seed. Seed endophytes can enter the seed from multiple pathways and are known to exist within all the different seed tissue types. But the role of seed endophytes in their host plant and in the ecosystem is not well established, especially within plants of natural ecosystems. Within some agricultural species, it has been shown seed endophytes can play a role in breaking seed dormancy and improving rates of germination. They can also play a role in root development through the production of auxins and ACC deaminase, although they can also exist as latent pathogens and saprotrophs, or decomposers, causing disease and decay in the host plant. And so I thought seed endophytes may play a role in addressing seed-based restoration issues, where there are often low rates of germination in the field, with some seedlings even failing to establish, and problems with seed decay and spoilage whilst the seed is in the soil seed bank. So, are seed microbes currently used in restoration within Australia? The short answer is not really, Increasingly, microbial diversity is being incorporated into restoration, but here the focus is being the monitoring of soil and root microbiomes. Seed biopriming techniques commonly used in agriculture are being investigated within Australia for restoration applications, but often soil microbes, not those from seed, are used. And there is work on seed banks as reservoirs of seed microbial diversity. And here there's a focus on wild crop relatives microbiomes, but this work is not based within Australia. And so, with a real gap in the integration of seed microbiomes in restoration and conservation within Australia, my research started out with a few simple questions. Do native Australian seeds have endophytes? And secondly, if they do, what species are there, and are they similar between different host species? So during my honours year, I collected seed from Banksia ericifolia and Banksia serrata from 4 sites in the Sydney region where they co-occurred. And for my PhD, I've collected seed from the grasses Themata triandra and Microlina stopoides from around 50 sites within the ACT and along the east coast of New South Wales. To isolate and identify the fungi and bacteria from the seed, I surface sterilise and macerate the seed, and the homogenate is plated onto growth media and incubated. From each pure culture, I extract the DNA and undertake PCR and Sanger sequencing. And the resultant DNA sequences allow for me to use phylogenetics to identify what species of fungi and bacteria are present. So excitingly, there was a diverse microbial community within the seed of all hosts. These column graphs show the breakup of the different fungal genera within the 2 Banksia's seed, from each host from each of the 4 sites. Here, each colour that is different represents a different fungal genus. The seed microbial community was really dominated by 1 or 2 highly abundant genera at each site, and many rare ones. And what was really interesting here, that even though these 2 Banksia species were sampled from the same sites, they house very different seed fungi. Ericifolia was dominated by members of the genus Penicillium, with this being dominant at every site. Whereas Banksia serrata was always dominated by a rare fungal genus called Banksiomyces. With the seed fungi of Themata and Microlina, I'm now seeing a much more even mix of genera present. There is not really one dominant fungal genus that's present at each site. We see that Themata is not always dominated by the same fungal genus, and here we see the Kosciuszko population dominated by Penicillium, whereas the Jarvis Bay site is dominated by Fusarium. What we are seeing for Microlina is a similar picture to what we were seeing with the Banksias, in that each site is dominated by the same fungal genus, in this case it's Alternaria. For the bacteria within Themata and Microlina seed, overall they are less diverse than the fungi, but we are seeing the same pattern, with the Themata sites differing in the dominant genus that's present, whereas the Microlina sites are always dominated by Pantoea. And another way to interpret these results is that Pantoea is always dominant and the Kosciuszko site is just an outlier. And I'm currently expanding on this data set, which will give us further insight into the spatial and host patterns that we are exploring here. The next question I was interested in was, what is the role of the seed microbiome in the ecosystem and within their host plants? And so I used literature records and fungal trophic mode databases to explore this a little further. Here, this phylogeny shows the relationships between the fungi isolated from the seed of the 2 Banksias. Here, plant pathogens are shown with a blue dot, plant endophytes are in pink, and decomposers are in orange. Interestingly, most endophytes are isolated were reported in the literature as being multitrophic, and this has been documented before, with endophytes becoming pathogenic on their host during times of stress or becoming decomposers after their host senesces. A similar pattern was shown with the seed fungi of the grass seeds, with many of the fungal species belonging to all 3 trophic modes, with a lot also being recorded as being both endophytic and decomposers. And so I started to conclude that the role that seed endophytes play within the ecosystem may actually be multifaceted and contextual. I also explored the role that endophytes play within their host plant. Here I investigated whether the fungi had been recorded as plant pathogens or being recorded to be able to promote plant growth. I found that many seed fungi are isolated, such as Alternaria, Coleoptricum, Epicoccum, and Penicillium, were being recorded as both plant pathogens and growth promoters, with this varying depending on the particular strain and the environmental context. I also found a similar story for many of the seed bacteria. I had a few Pantoea species in particular that were recorded as being both beneficial and pathogenic on their hosts. And so how can this knowledge be integrated into restoration and conservation? Seed collections may provide an opportunity to capture not just the genetic diversity, but also the microbial diversity of a plant species. And our preliminary data suggests that even co-occurring plant species may each house a unique seed microbiome, and different populations of a host species may have a similar highly abundant microbial species present. Capturing seed microbiomes through seed bank storage may include viability checks of the seed, but also its microbiome. Our research highlights that the role of seed microbes in the host and ecosystem is complex and perhaps dependent on the particular strain present and other environmental factors. But what is clear is that there is a hidden diversity within our native seed that could contribute to the conservation and restoration of our native plants. Thank you.
Hi, my name is Maurice van der Merwe, and we were interested in investigating the diversity and dynamics of fungal endophytes found within the seeds of native Banksia. Firstly, we have to start by asking the question of what is an endophyte? Well, an endophyte is an organism, a fungi or bacteria, that lives asymptomatically inside a plant for at least part of their life cycle. They have been found to be both pathogenic and beneficial to the host, and particularly within seeds have shown to aid in stress tolerance, root growth, provision of nutrients and water, as well as aiding in nutrient cycling. Understanding the beneficial role that these microorganisms play within seeds, we were particularly interested in the factors that may influence the structure of these communities within native Banksia. We therefore examined whether host species or site influences these communities. To achieve this, we used the study system of co-occurring Banksia ericifolia and Banksia serrata. Both of these species hold their seeds in canopy-held seed banks, which are predominantly released after a fire. These species are an abundant and important species within ecosystems and are frequently included in revegetation and conservation programs, where seed germination and establishment is particularly important. The seeds were collected from 4 national parks in the Greater Sydney region, where both species co-occur. At each of these sites, 30 infractescences were collected from the canopy of each of the hosts. From each of these infractescences, 10 seeds were removed, totaling 1,200 seeds per host. These seeds were surface sterilized and plated onto growth media and incubated. On the plates where fungal cultures grew, DNA was extracted from the mycelium. PCR was used to amplify the ITS region of nuclear DNA. These fragments were sequenced and clustered into operational taxonomic units, or OTUs, based upon 97% similarity, commonly used as a proxy for species in fungal diversity studies. Phylogenetic tree construction was used to identify the endophyte species present. Our Permanova results indicated that at every site, the community in Banksia serrata was different to that in ericifolia, regardless of data transformation. This result is visualized in the NMDS. Each of the ellipses represent the samples from site of each host. Ellipses in shade of red represent communities in ericifolia, and those in blue show the communities of serrata. Ellipses closer together are communities more similar in composition. Samples from each host form 2 distinct clusters. Banksia ericifolia always had the same few penicillium species present, which is highlighted in the red boxes. These penicillium species are both known as latent pathogens and saprotrophs, producing mycotoxins and antibiotics toxic to mammals and have been associated with increasing root growth in host plants. Whereas, in comparison, Banksia serrata always had Banksia myces species present. This fungal species has only been identified as a saprotroph found on dead Banksia cones, as shown in the images here. This species was only ever found in Banksia serrata. But what does this mean for conservation in the field? If we are trying to conserve microbial diversity of an ecosystem, potentially each host species may house a community, and this diversity may be structured at a species rather than a genus level. If we are trying to introduce microbial diversity into an ecosystem, even hosts of the same genus may have a unique contribution. For both hosts, the endophyte communities differed from site to site, with this trend mostly driven by rare species within each community. In this NMDS, sites that do not share a letter are significantly different in composition. Therefore, if you are wanting to collect seed of a host that is also representative of its endophyte diversity, you will need to collect from multiple sites of that host. So, in isolating a diverse range of fungal endophytes from Banksia seed, it is still not completely clear the full range of ecological filters that shape the assembly in these communities, although it appears that the community is formed by both site and host-related factors. Interestingly, there were a few highly abundant species within each host that were consistently isolated from geographically distinct sites, which may play an important role within their hosts. We were then interested in furthering this knowledge by asking whether these microbial communities are influenced over time and space. Using a single host species, Banksia ericifolia, we investigated whether seed age and urbanization influence microbial diversity and composition. Firstly, we examined seed age and categorized the seeds into old, which were seeds that had been held in the seed canopy for over 8 years, versus young seed that have been held for less than 4 years. Seeds were collected from 2 national parks in the Greater Sydney region, which contain trees older than 25 years old based upon the last time since fire. At each site, 30 old and 30 young infractescences were collected. From each of these infractescences, 10 seeds were removed, totaling 600 seeds per site. We then followed the same methodology as our previous study to identify the fungal endophytes present, although we then blasted the sequences. Permanova results indicated that the endophyte communities differed with age, and the differences were site-specific. The assemblages of endophytes in seeds from older cones were distinct from the assemblages in younger cones. We also found that Penicillium citrionnigrum was predominantly found within older individuals, which is highlighted in the red boxes, in comparison to a Neophysiococcus species, which was most abundant in younger individuals, highlighted in the yellow boxes. The variation in the abundance of common shared endophytic species provides insight into the potential role that these organisms play as the seed ages, while also providing insight into the colonization pathways that these microorganisms use, with our results showing patterns that suggest a maternal transmission of fungal endophytes into the young seed. Secondly, we investigated the influence of urbanization on the diversity and composition of seed fungal endophytes. 2 remnant vegetation sites located within the city of Sydney was chosen. At each site, 30 young infractescences were collected, with a total of 300 seeds per site. We then followed the same procedure as before to identify the species present. When comparing the results to the young cones collected at the native sites, we found that the seed fungal endophyte community did not change significantly. Across both natural and urban sites, the seed fungal endophyte communities consisted of some highly abundant species and some rare species, with Banksia myces being the most abundant species found within the urban seeds. So, the variety of different fungal species identified within both studies suggests that these species undertake a range of different functions which need to be identified. Such knowledge will be important for the storage of seeds for restoration activities. At present, plants in urban national parks appear to be very similar to those in more natural areas, although the function of a suite of unique endophytes in urban areas needs to be further investigated.
Hi there, thanks for listening in. So today I'm going to be talking about smoke and germination in unburnt vegetation. And it's an idea I've been playing with. I think I saw notes in my old notebook from 2009 where I first started thinking about this idea. So good to finally see the light of day. So the majority of species recruit only after fire in fire-prone systems. And that's partly because in unburnt habitat, it's not conducive for seedlings to successfully grow and recruit. And so species really want to make sure that they germinate into that post-fire environment. And you usually see that by a big flush of emergence, like in the picture here. Seed banks are maintained by these species. Many of them have soil stored seed banks, and that maintains seeds in the system in between fires. And it's dormancy and germination cues that ensure that emergence occurs at the right time. So some of the dormancy classes, probably many of you are aware of, well, the two main ones at least that are important, I think, in fire-prone systems are physically dormant species and physiologically dormant species. Physically dormant species have that hard seed coat and mostly overcome that dormancy by a heat shock driven by the passage of fire. The physiologically dormant species, on the other hand, generally require some sort of seasonal temperature variation to overcome dormancy, and they also have a smoke cue. They're the ones that respond and have that germination cue to smoke, and that smoke is usually only in very small concentrations that's all that's required. Now the thing is, smoke travels far and wide, so a single small prescribed fire can generate quite a lot of smoke. A bit of work by Owen Price down at Wollongong and colleagues down there found that a small, very small 50-hectare prescribed fire has a smoke plume that can extend around 9 kilometres, and a 700-hectare fire, the plume extended around 14 kilometres. But as we know, climate's changing, and it's producing conditions that are conducive to really big fires, something we're calling megafires. And so that's going to have a big impact on what's happening as far as smoke is concerned. So we all know the big bushfires of 2019 fire season meant that entire ecosystems were affected. And you can see on the map there, the brown colours are the extent of the bushfires just in New South Wales alone. Around 5.5 million hectares burnt. It's a large part of national parks and state forests, and many threatened fauna and flora species were in that fire footprint. But perhaps not as much looked at is that in many of the unburnt areas, smoke covered the landscape, sometimes for weeks at a time. And following these fires, there was drought-breaking rains, and we know that many of the soluble compounds in smoke that break dormancy or promote germination, I should say, in physiologically dormant species are water-soluble. So if there's smoke all over the unburnt areas in these systems, what is it? What's the mechanism that's preventing this suicidal germination of seedlings, of seeds into unburnt vegetation? So to look at that, we used really what's one of the perfect case study species for this kind of experiment is Barrerro floribunda. It's endemic to the Sydney region. It has physiologically dormant seeds that form as a soil stored seed bank. And it's a smoke responsive species. So we had a look after a fire in the habitat where Barrerro floribunda occurs to see just, is this having this big response to fire, and do we see a similar increase in the amount of seedlings in the unburnt area? So you can see from that figure that in the burnt area, there's a really big number of seedlings that are emerging from seeds in the seed bank, whereas in the unburnt area, you really don't get much response at all. So this is following the pattern that we sort of expect and see. The other thing with Barrerro floribunda is that it has maximum germination in winter or autumn temperatures, and it's obligate to smoke. In other words, you can see in that bottom figure there, the two black bars on the right there, groups of bars have been germinated at autumn and winter temperatures, and those black bars there are the peak germination that occurred after a smoke application. So it's an obligate smoke responder, and it likes those cooler temperatures. So the main question we asked in this study was, how do the many species with physiologically dormant seeds prevent germination in an unburnt but smoke-filled landscape. And we hypothesized that because many physiologically dormant species have a seasonal requirement for breaking dormancy and promoting germination, there's likely to be something to do with the temperature they experience in the burnt or unburnt habitats. So we went out and measured soil temperatures both under canopy, so in unburnt areas, but also in open areas where the canopy had been removed by fire. And we could see that, yes, there's quite a difference. If you've got a nice intact canopy that hasn't been burnt, in typical summer conditions, you're getting around 25 in the day and 18 at night. And in winter conditions, you're getting around 14 degrees in the day and 11 at night. And these are much narrower diurnal ranges than you can see in the above corresponding open areas that have been burnt. And so no canopy providing shade. So we ran a germination experiment to see whether or not these varying diurnal ranges that aren't that big really, the difference between, in the winter ones, between 16.11 under a canopy and 17.6 in the open. It's not a massive difference. But we ran a germination experiment under these combination of temperatures to see if there was in fact a response. And you could see that Barrerro showed a clear effect. Firstly, germinating mainly at winter temperatures, but also 3 times less germination in temperatures produced under canopy compared to those open temperatures where the canopy had been removed by fire. So pretty clearly we can see that it's the diurnal temperature ranges that are helping to control that suicidal germination into unburnt areas, even though the smoke cue is present. But I think we need to study more of how diurnal temperature ranges vary, and similarly how species respond to those diurnal variations. And we also need to consider how climate is going to change in the future and how these shifts in diurnal temperatures may also change along with that. Thanks to everyone.
Hi everyone, greetings from Sarja Seed Bank and Adrienne. I'm Sanjay with my colleague Dr. Hatem, Mr. Tamar and Ms. Asma. The topic of today's presentation is entitled Seed Conservation and Germination Response of Important Native Grasses from Arid Deserts of the United Arab Emirates. Native grasses have the potential to be good candidates for fodder production and restoration of degraded rangelands because of their palatability, nutritive value, and adaptation to local environmental conditions. The conservation of seeds in seed banks has become a central pillar in current global conservation efforts, and seed banks are becoming resources of vital importance for preserving genetic and species diversity for future use. Seed viability differs between species and within a species in response to the storage. The aim of this study was to investigate the germinability of freshly matured and 3-year stored seeds in seed bank. 8 accession of freshly collected seeds and 3-year stored seeds of 6 grasses were used in this study. Germination tests were performed using standard seed germination protocol for testing in the seed banks. In all cases, seeds were germinated in a plant growth chamber with 12-hour light, 12-hour dark photoperiod, and continuous darkness at 20-30 temperature, as we found this optimal for these species. The germination data were analyzed and responses were compared. The results shows that freshly matured seeds showed higher germination and velocity. Synchronous accession to Synthropodia, Penicitum, and Sporobolus maintained germination percentage and velocity during storage. Also, there was improvement in germinability during storage in synchronous accession A1 and Lacerus accession A1. So the dry storage might break dormancy through after ripening process for the species mainly having non-deep physiological dormancy. The storage induced a dark requirement with an increase in germination percentage, indicating that storage might change phytochromic sensitivity in the dark. The low germination reported for Lacerus accession 2 can be related to maternal habitat and moldiness of seeds during the germination process. For Stiphagrostis, the results indicating that storage might induce changes in temperature requirement in order to break dormancy. In general, the results obtained indicated that the storage conditions in the seed banks are suitable for the preservation of native grasses seeds. The ability to maintain viable seeds of native grasses in storage and understanding of the germination behavior is essential for improving their future uses. Future work include periodic viability monitoring and predicting seed storage life. Thank you for your attention.
Hello everyone, my name is Arina and today I will be presenting a brief summary of one of our studies that was conducted on narrow-leaf lupin seeds. Lupin is a legume and it's a promising source of plant-based protein and fiber. The main proteins in lupin seeds are the conglutins, which are classified into 4 major groups, the alpha, beta, gamma, and delta conglutin families. Many nutritional and pharmaceutical properties are reported from these proteins. For instance, the gamma conglutins have blood glucose lowering effects, and the beta conglutins, which are the known allergens from lupin, have cholesterol lowering, antioxidant, and anti-inflammatory activities. This proteomics workflow was used to measure the conglutin proteins and comparing them across genetically diverse narrow-leaf lupin varieties. Firstly, the proteins were extracted from lupin flower, and then they were subjected to tryptic digestion. The resulted peptides were separated by liquid chromatography and characterized by mass spectrometry. This resulted in the identification of proteins from lupin. Then in the quantitative phase of the study, the peptides specific to the conglutin proteins were selected, and they were included in a targeted quantitative method in order to selectively quantify the conglutin proteins across 46 narrow-leaf lupin varieties. Within the major conglutin families, there are the protein subfamilies, and overall 16 conglutin sequences are reported from narrow-leaf lupin. These color-coded blocks actually are the specific peptides mapped back on the protein sequences, and these were used as the marker peptides in the targeted method to differentiate and quantify these 16 conglutin sequences. Then the peak area of the specific peptides of an individual protein were summed, and are represented in these bar charts, which are comparing the conglutin profiles of the analyzed lines. As you can see, the levels of the alpha conglutins were quite consistent across these genotypes, and so as the gamma conglutins, with an exception of some of the lines that they had higher content of gamma conglutin. However, the variability was much larger in beta and delta conglutins. If we have a closer look at the beta conglutins, you will see that interestingly, some of the domesticated lines that are showing distinctly different beta conglutin profiles. So using mass spectrometry, we were able to quantify some of the nutritional and anti-nutritional proteins from lupin. We identified the distinct differences between the wild and domesticated varieties at conglutin level. And these are all useful information for selective breeding programs and development of varieties with quality protein complement. Thank you for your attention.
Hi everyone, I'm Ganesha, a scientific officer from the Australian Plant Bank. Today I'm going to talk about the seed dormancy breaking of Australian rainfall species based on a case study that we've done on Acronychia, a genus commonly known to have difficulties in germination. Firstly, I would like to pay my respect to the Dharawal people, traditional custodian of the land on where I am presenting this talk today. I also would like to acknowledge all the funding bodies of Rainforest Seed Conservation Project at the Royal Botanic Gardens and Domain Trust for supporting this research. As most of you know, seed dormancy is an innate seed trait that controls seed germination. Ecologically, it is important because it can prevent seed germination in unfavorable conditions where subsequent seedling establishment can fail. However, in conservation perspective, dormancy is problematic. As an example, seed banking involves drying and storing seed in sub-zero temperatures. Testing species suitability for drying and freezing involves a series of germination tests. Performing such germination tests are difficult when dormancy breaking technique for targeted species is unknown. Our aim was to investigate a dormancy breaking technique for Acronychia. We tested 3 species, Acronychia imperforata, Acronychia levis, and Acronychia oblongifolia. We used 4 dormancy breaking treatments: full seed coat removal, here decoating, use of gibberellic acid, scarification on a place opposite to the radical emergence point, and scarification on radical emergence point. Highest germination was observed in decoating and scarification near the emergence point treatments. Though the scarification increased germination, it was low when seed coats were scarified on a place opposite to the radical emergence point. The imbibition test also confirms seed coat permeability. Then why the clearance near radical emergence point is essential for germination? This could indicate an embryo's inability to push through the seed coat, which is a characteristic of underdeveloped embryos. However, in Rutaceae species are known to have developed embryos, and embryo to seed length ratios also confirm the presence of developed embryo. Inability of developed embryos to push through the seed coat is found in seeds with non-deep physiological dormancy. Therefore, we confirm the presence of non-deep physiological dormancy in genus Acronychia using our results and scarification near the radical emergence point as an effective technique for breaking dormancy.
Hey everyone, thank you for some wonderful talks. We'll just wait for all of the speakers who are going to join us to appear. Very good. That brings us to the end of our final session before lunch, and we've got a few minutes for some questions about those talks. I'm going to start with Jose. Jose, there's a question that you've answered online, but can you was there anything else you wanted to add about the impact of the modification on germination or establishment in cowpeas?
Yeah. The question was if our transgene, our new protein we're introducing in cowpeas, have any impact in germination establishment, as you said. And the answer is no. We haven't seen any penalty of having this new protein in. We are expressing it at the levels found already in beans using the native promoter. So it should be rare to see any issue such as that.
Thanks. So just another question. This one's from me. This alpha amylase looks like a pretty useful protein. What do you understand about the phylogeny of it? Is it common in other seeds or?
Yeah. No. There are many different kinds of alpha amylases in many different species, from cereals to legumes, as I was watching here. And it seems what is very interesting is how the insects are in an arms race to become resistant to those enzymes that stop them to feed on the plant. So in the example I was using in my presentation, you can see how the Americans coleopters have overcome that alpha amylase inhibitor from American beans. But in Africa, the coleoptera are still sensitive. So it's interesting how you can now try to move across evolution and move them from one continent to the other.
Thanks, Jose. I might go to Sean next. Thanks for your talk, Sean. I was impressed at your technique. Can you comment on the sort of limitations of the potential to extract seeds from soil, say in communities that are more diverse than the one that you were working in?
Sure. Can you hear me okay?
Mm-hmm.
Obviously, this method only works when you're looking at large seeds. So, it works fine for acacia because they're visibly distinguishable from other things that you would find in a soil sample. It'd be no good for anything that was, like, say, 10 times smaller than that. getting to the, yeah, I mean, let's say you were interested in a community that had multiple species of the one genus, where you could recognize at genus level what the seed was, but that you wouldn't necessarily know what it was at a species level. That's another factor. I actually managed to distinguish probably 2 or 3 different species within the samples that I found. Acacia melanoxylon and Acacia obliquonervia stood out as being very physically different when you look at the seed versus Acacia delbata, my target species. So I was able to actually pull them out and exclude them from the experiment. But yeah, it's the method I've developed is really for a single species study, but you could potentially expand it to do other species.
Thanks, Sean. Question for you, Mark. Lydia asks if you have any thoughts on whether the relationship between the diurnal temperature range would be linear, the bigger the day-night difference, the more germination, or a curve. And I'll add a question to that, which is, do you have any sense of whether it might be either the top or the bottom of the range that's triggering the effect, or do you really think it's the range itself?
I mean, the short answer is I'm not sure, but my gut feeling is that it's probably curved and possibly there's just a threshold. And I think it would probably be closer to the top of the range, as in, sorry, the greater the range. And I partly think that for some species there has to be, and like the Barrerro, perhaps there has to be a lower temperature that has to be hit for germination to occur, and that might be part of that sort of threshold trigger. But more broadly, I think what's interesting is that it may be that this diurnal sort of limitation is stronger in some species than others. And if that's the case, you might find sort of functional groups of species that may be more susceptible to this sort of smoke covering the landscape than others. And I think that's sort of a real impetus to have a look at what other species do in this situation.
Thanks. Sean had a question for you as well, as to whether you think that the age of smoke might matter in this scenario.
The what, sorry? The
age. Sean, you want to ask that question since you're here?
Yeah, this is, I just know, because I used to be an atmospheric scientist, I know that smoke actually does age. You have various chemical reactions take place over the course of some hours. And some of those are like photochemical reactions. They can make ozone and stuff like that. So I was just wondering whether there's been any studies looking at freshly generated smoke versus smoke that's been sort of circulating in the atmosphere for some time.
Yeah, I'm not entirely sure. There's probably people here better qualified to answer that than me. But again, if you get rainfall occurring quite a long time after a fire, it doesn't matter whether it's been in the fire field, under the fire footprint or beyond. Usually it's rain that then washes those carrageenolides and other things down through the substrate. So yeah, I don't know. Sounds like an interesting area to have a look at, but yeah, that's probably as far as I know, I think.
Thanks.
Thanks, Mark. Thanks, Sean. I've saved Alison and Marie's for last because there are several questions for you both, and I think actually I'm going to pitch them all to both of you, and you can just start to take turns. Kelly Gowan asked whether you thought about what the definition of a healthy soil, seed microbiome might be, and what you think the potential consequences could be of translocating microbiome components, say in a restoration setting might be. I'll start with Alison.
Sure. Thanks, Adrienne, and thanks for the question. Can you hear me clearly?
Yep.
Yep, super. I suppose I'll answer in relation to the question from Kelly in respect to soil first. So defining a healthy soil microbiome or a healthy seed microbiome, I think is particularly challenging, and I feel like there has been a little bit more work done with this in respect to soil microbiomes. And from the literature, what I can kind of gain is that healthy is kind of defined as one that is most similar to a natural reference site. So say if you're trying to restore a site that is of a certain vegetation complex, and it has a certain soil microbiome, and you're aiming to restore your degraded site to that point, you'd be aiming for a soil microbiome that is similar to that. But whether it's similar in microbial species composition, whether it's similar in functional diversity, I think is kind of various ways that it's been looked at. But there's no definition of a microbiome that is healthy and one that is unhealthy in the same way that we have with the human gut or something like that. And I think it is, from the research, kind of more site-specific and vegetation complex-specific. And with seeds, yeah, we don't have any understanding of what a healthy seed microbiome is yet. I think especially for native plants, we're just scratching the surface of what is there and what role may they play. And that's still, yeah, very early days so far.
Maurice, did you want to add anything to that? Okay, well, here's one for you then. Can you chat a little bit about how you think seed banking might be affecting microbiomes, and how you think we might need to alter our approach?
Yeah, I think that's a great question. I think, like we said, there's not actually that much research into the seed microbiome particularly, and I guess what we're interested in is how kind of almost a role that these microorganisms may play within the seeds, and over time as we're storing the seeds, whether, some seeds lose germination capacity and whether the actual endophytes that we might find in these differ as well, and that can kind of give us an idea of the roles that they may play, and how we can then preserve them for when we do kind of rehabilitation of sites and ensuring that we're kind of encompassing the whole seed in there as well. And I don't know if Alison wants to add anything to that as well.
Alison?
Yeah, I think in terms of using seed banks, I suppose there's a lot of challenges with collecting representative populations of a particular plant species and trying to capture the genetic diversity of that species. And I think just kind of opening up the question or the idea of, okay, well, let's add another challenge, and how about use seed banks as a way to capture the microbial diversity that's within seeds. So yeah, make things extra hard for ourselves, but it might be an opportunity to provide a way to look at conserving microbial diversity, because it is hard to conserve because you can't see it, and seeds might provide a way of capturing and storing that for future work and for capturing ecosystem diversity overall.
Thank you. I think that pretty much brings us to the end of the questions that we had in the Q&A. Do any of my speakers have a closing question for any of my other speakers? Okay, I have one final one with the microbiome stuff. Curious just to know what you think our priorities should be. It's such a new area. How do we prioritize research in this space? And for people who are thinking of going in this direction, what would the two of you suggest they focus on first?
I might quickly jump in whilst Maurice is gathering. I would say the first step is understanding the diversity of what microbial species are present within native plants, in particular within the Australasian region. Our understanding of seed endophytes comes predominantly from agricultural species. So I think identifying what species are in natural ecosystem plants, and because the processes and the methodology to be able to do that and understand that are being used in other systems within foliar endophytes and things like that. So I think that would be a good starting point. And then from there, understanding potential function, I think the experiments and the techniques are much less well understood within seed endophytes there.
Thanks. Maurice, did you want to add anything to that?
Yeah, no, I think what Alison says is pretty on point. I think, there, like she said, there's a lot of research into agricultural species, but very little into the native plants. And I think, just finding out what there is at the moment, because we say there's so very little, like there's such little research into the seed microbiome, particularly of native species. So I think the starting point is just going, well, what is there? And then I think kind of, well, how do they get there and why are they there? All those really important questions. And there's so many different questions you could answer. But I think a good starting point is just going, well, what is there and how, do they differ between species and plants and why is that? But yeah, no, I think Alison's pretty spot on there.
I suspect there won't be many more talks in this space next time the conference is held. It's a really exciting area. We also had a few of the 3-minute speakers who haven't been able to join us on the talk on the screen for the Q&A. But if you have questions for them, please go ahead and pop them in the hub using the portal. So thanks everyone for a fabulous morning of talks. We have a lunch break now, and we'll look forward to seeing you all again at 2 o'clock. Eastern Australian time, or whatever time that might be for you, wherever you are in the world. See you later.